Okay, so just standard reminders on these points due this Sunday. The concept check that was originally due this past weekend because we didn't through mutation last time was bumped to being due Wednesday of this week. And then the mini project is due next Wednesday. That's the only mini project that is not dropped. So that is implementing a very simple genetic algorithm that is will optimize a mathematical function, analytical function that's very, you know what the optimum are, but it has lots of local optima and constraints. So it gives you an opportunity to see how it moves through the search space, and then you can explore different types of operators. So the goals for today then, we're definitely going to wrap up the GA and discuss how these 5 hyperparameters plus selection pressure work together to set up the different features of the algorithm's performance. And if we manage to get through that, then we'll start Unit 2, where we introduce evolutionary computing and some other frameworks for thinking about these evolutionary algorithms that may look a little bit more like conventional machine learning, and then we'll transition to evolutionary programming, where you actually start doing more creative things with these algorithms, where you're not optimizing a function, but you're actually having them write code for you, for example. So that's kind of the beginning there of Unit 2. But going back to the end of this unit, so just as a reminder, I've shown you 2 basic structures of a GA, the GA from the book and the GA from MATLAB. If you'd like to see the pseudocode of the GA from the book, I've made that a little easier to find. So if you go onto the Canvas site and go to modules and under Unit 1, there's a link right there. You can actually see the pseudocode for the GA from this book. The big difference between these 2 GAs is how they manage their population structure, but the general things that I'm going to say about the parameters when you crank one up, when you crank one down, is going to apply to most all GAs, even if they have slightly different population structures. The quantitative results change based on how they set up their population, but the qualitative results don't really. So there are sometimes pragmatic implementation reasons for why why the MathWorks engineers decided to set up their GA based on this model versus why the folks who wrote the textbook decided to do this one. So there could be pedagogical reasons why it's simpler to start with this one, and there could be more modular, scalable reasons why you might go with this one in practice. I will tell you that most practical GAs will have formal elitism, so that's kind of a big thing that the MATLAB one has, where they've kind of left it out of the book one. But on the other hand, I've actually given online a couple of different textbooks. This book, as well as a book by Passano, and the Passano book, when they introduced the genetic algorithm, also uses basically this structure. So this is, I think, a common one you see in textbooks, and this is a common one you'll see in practice. So just to put those up there, key thing in each GA, these 3 operators really drive everything. So those are the 3 things that you're deciding amongst. And so the selection operator we talked about last time. So here's your basic GA structure here. And so you've got these incoming population from the previous generation. You've got to figure out who the parents are that seed the next offspring in the next generation. And that selection operator is what does that. So we talked about 3 different selection operators. Fitness proportionate selection, which is a very popular one. Tends to be kind of an intuitive one where people's minds go. Ranking selection and tournament selection. I would say the most popular selection operators that you'll see out there that'll come pre-built are usually fitness proportion selection and tournament selection. Turns out a lot of the things that you can get out of ranking selection, you can also get out of tournament selection. So that ends up becoming kind of more popular. So that's what we talked about with those. And then we then moved to recombination. And this is kind of where we're going to need to go back and look at, because where we got cut off before. But after you select your parents, then you need to then populate the offspring pool. And so you either are going to do that by creating offspring by recombining individuals as the parents, or by mutating the parents, or by doing both. So we're focusing on the first part of that here, this recombination. So sometimes it'll be called sex, sometimes it'll be called crossover, and then more often than not, it's just called recombination. But the operator is almost always called the crossover operator. And there's different ways to do crossover. And so generally speaking, the crossover operator is doing something like this single point crossover, where you've got the genotype of one parent, the genotype of another parent, you pick a point, and then anything after that point gets swapped. So then you have parent one ends up showing up in both offspring one and offspring two. First half of parent one is an offspring one, second half of offspring one, or parent one is an offspring two, and vice versa. So first half of parent two shows up in offspring two, second half of parent two shows up in offspring one. And that's what we call single point crossover. And so that's what we talked about last time, is that, exactly what I just said there, this crossover point. So basically you look at your parent pool, and there's some process by selecting two individuals. The details of how you select those two individuals at random can vary, but they are kind of inconsequential. So in one of the textbooks, like the Passano textbook, I think the way they do it is there's a probability of crossover that gets applied to every single parent, and you basically get tagged. Do you get selected for crossover or you not? And so every parent gets either a 1 or a 0, whether you're going to get crossover or not. And then they look at the subset that got tagged for crossover, and then they randomly choose among those. And then from there, then they'll take two of those, then create two offspring and put them in the pool. Now you could do that a different way. You could draw two from the whole parent pool and then flip your crossover coin and say, these two have met. Do they engage in recombination? Do they have sex? And if they don't, then you just put them back in the pool. Maybe if they don't, you just copy them directly into without crossing over at all. I mean, there's different choices you can make, but point is, all of these algorithms will have some sort of crossover rate or crossover fraction. So the way MATLAB does it is it has these M minus E spots left over that it needs to populate offspring. And you tell it, I want 30% of those to be crossed over. And so it just says, all right, I need to fill up M minus E times 30%, and then it just populates those. And that's how it does that. So in all of these different ways, if you crank that crossover probability up, you're going to get more offspring that are mixtures of two parents. And if you crank it down, you'll get more offspring that are, that are not mixtures of two parents and probably just mutated versions of one. So and now that crossover probability, as we'll talk about here in a moment, you often will have that high early in the algorithm. But as we'll see, as we started to work out numerical examples last time, as your diversity goes away, as all the parents start looking similar to each other, crossover becomes kind of a computational waste, because the crossovers don't end up doing anything, and so you spent this time doing this crossover, and you end up getting offspring that don't really look any different than the parents. So often, you just turn off crossover so you might run 100 iterations, generate a population that has benefited or at least has been affected by crossover, take that population, make it the initial condition of a new GA that runs 100 iterations without crossover. And so that's like, something common you could do. Or what some GA algorithms do is they decay the crossover probability over time for you. And they give you another hyperparameter that sets that decay rate. So you set the initial crossover rate, and then over time, it gets lower and lower and lower automatically. So that's another way you could do that. So there's just a little bit of flexibility. But let's figure out, different types of crossover and what crossover does. So single point crossover, again, you just pick a random point. You've got two parents, they've been selected for crossover. It's going to happen. You just don't know where in the chromosome that crossover point is going to happen. So the two parents come together, you draw a random gene that is the sort of like marks the last gene that doesn't get crossed over, and then anything after that, then you just swap things so that offspring 2 gets parent 1 and offspring 1 gets parent 2. So that's our 1-point crossover. Likewise, you could configure that as 2-point crossover. So that means you want more swapping. And then you draw 2 points, and then anything in between the 2 fence posts, those 2 genes get swapped, everything else stays in the same spot. And you can play that game over and over again. And the more points of crossover, the more opportunities you have to break up what's called linkage. And the idea behind linkage is, you might find, as an example, let's say this is a design for a car, common example that I talk about. So this gene might be how many wheels you have. This gene might be the shape of your steering wheel. This gene might be the type of fuel octane you use, and so on and so forth. Well, if you've got the fuel octane right next to the number of wheels, then if you happen to have a genotype that has a really high fitness number of wheels, then if that's really driving, so to speak, the fitness of this car, then that may carry all of these other alleles that are next to it with it. So if this one allele happens to just really have a significant impact on fitness, then these alleles might ride the coattails of this allele's impact on this individual's fitness. And so that means that you're masking selective pressure on these alleles because they're just along for the ride. So what crossover does is it allows you to explore other types of, say, 4-wheeled vehicles that have different fuel octane, because it sort of helps ensure that periodically you're going to break up these genes, you're going to swap these genes, and now you'll end up getting an offspring that has the same number of wheels, but maybe uses a different type of fuel. And so that's the idea of crossover, is it breaks up so-called linkage. And that can allow for discovering new solutions that otherwise would have been hard to find, because this string-based representation creates a coupling between alleles that may not have anything to do with each other. That's the point of crossover. And so to really mix things up, you could then go to uniform crossover, where you don't select crossover points. You have a crossover rate per gene. And so you got 2 parents, and then you just walk down through every gene of every parent, and then you say, all right, so for these 2, which do we keep them in the current order, or do we swap? And then so you could say, we're going to swap this one, we're going to keep these the same, we'll swap this one, keep these the same, and so on. And then you can sort of shuffle all of those mappings. So those are our point-wise crossover operators. Any questions about that? Yeah.
So I have a question around the number of parents. So in biological sense, it's okay with the crossover have 2 parents. But while we are doing optimization and we need sample, so the crossover is it possible that the crossover might happen with more than 2 parents with a single offspring?
It's a great question. The question is, can you define a crossover operator with more than 2 parents? In the standard GA, you can plug in any crossover operator you want, but there's an interface spec that's going to say you have to take 2 inputs and you have to produce 2 outputs. So for a GA, we're kind of limited to the 2 in, 2 out. Now there are other evolutionary algorithms. So at the beginning of Unit 2, I'm going to introduce what's so-called ES, or evolution strategies. Some of you may have heard of CMA-ES, covariance matrix adaptation evolution strategy. It's sort of a popular way to do searches, and that's one of examples of these. And in CMA-ES and other ES, there are crossover where you can define an arbitrary number of parents. Now you can say, well, how does that happen? We'll talk about those structures, but it requires a sort of a fundamentally different way of thinking about this. So for example, you could imagine I have a set of parents, and I need to produce 1 offspring. Well, that 1 offspring at every gene location draws randomly from the parents for the source of that gene. That would be one way to have an in parents in, one offspring out, version of that. Or in the arithmetic case, which is the next slide here, you could imagine having a weighted average of a bunch of these parents, and they could even be weighted by the fitness of those parents. And then you could have multiple in and one out. So typically with those solutions, when you have an arbitrary number of parents, you only produce one offspring, because the mechanisms there kind of don't make sense. Like, how do you go into two? It's kind of a weird thing. But if you have the same in and the same out, 2 to 2, then it works out okay. So the GA generally assumes a 2 to 2. So if you go into any GA and you plop in your own crossover operator, you're going to be forced to this interface spec. Other questions about this pointwise crossover? Okay, so you don't have to do crossover with by points. It's also possible to do a linear combination, or we might call a convex combination, or even a conical combination. So where you can draw a weight, which is a mixture of the two parents. And so this is, remember, this genotype here is, just as a reminder, let me use a different color, not red, so it's black. So as a reminder, x1 vector here is equal to x11, x12, all the way down to x1n, where n is our number of genes. And so we're doing this linear combination on every gene. So on every gene, you're doing alpha, which is just a scalar, times the gene from parent 1, plus 1 minus alpha times that same gene location from parent 2. Or I should say the allele. So you're doing alpha times the allele for parent 1 plus 1 minus alpha from the allele from parent 2. And you're mixing those together gene by gene by gene. So this is very fast, especially now we have GPUs, GPUs are built to do this. This is a little linear combination of two parents. As we'll see in these numerical examples, it achieves a lot of the same thing, especially if you allow these alphas to even vary more wildly than this. It's a way to sort of create mixtures of offspring that are more directed than a totally random search. Question?
Is this different from uniform?
In the previous case, I'm going to swap back for a second. In uniform crossover, this was discrete crossover in that you're literally swapping. So you're not mixing. So you're saying the offspring is either going to, I guess I should have made this, when I wrote these as Y's, these should probably be X's down here. But the idea here is that the offspring is either going to take the exact value from parent 1 or the exact value from parent 2. So at this location, this is like, number of wheels in the car. Well, parent 1 might have 3 and parent 2 might have 4. So offspring 1 is either going to have 3 or 4, but nothing else. Whereas over here, if you had offspring, parent 1 had 3 and parent 2 had 4, then if you're doing this, we're assuming that all of these genes are real numbers. And so the offspring might be 3.26, and then another offspring will be the other. So you're going to do you might say, I'm going to be 25% of whatever the parent 1 value is, plus 75% of whatever the parent 2 value is. And then I'm going to switch that, and I'll be 75% the parent 1 value plus 25% parent 2 value. So now you're going to be guaranteed to get something numerically in between the values of the two parents. That's why this has to be real value. So if I didn't write that in here, I'll just make a note. So note, we have to have real valued genes. So continuous. All right, questions about that approach? It's a little more numerically faster, it's more efficient. And when we go into evolution strategies, it's already brought that up, we find that they end up doing a lot more with this approach where they're a little more clever about how to draw this weight. Yep.
So we're gonna use both of them depending on the type of data.
It's up to you. So when you're configuring the GA, you might decide that for my GA, I'm going to do this type of crossover. And a lot of times it might just come down to speed. It's like I've got this huge vector. You know, this might be a 1,000-degree vector, a 1,000,000-degree vector, or something like that. And if you think about that, if you're trying to write code to do this, now you got to iterate down through there. You got to swap data from one thing to another. This just becomes computationally, ugly, right? I can't do this, it's not really an atomic operation. But here, again, especially now that we have GPUs that are made to do linear transforms, then this could be a big ugly, vector. But as long as it's a bunch of real values, I can do this linear transformation like that. So remember, in a GA, if you have a population of 1,000 individuals, every individual is a costly fitness evaluation. Every individual has to evaluate your optimization objective once per generation. So that's 1,000 optimization objectives. So like, again, I mentioned I do a water quality or a wetland placement. I have a research project where we have to optimize these large river basins based on where to put wetlands and things like that. And that hydrologic simulation, every time I make a choice of where to put wetlands in this basin, takes 10 minutes to calculate my F. So it takes 10 minutes per individual to calculate fitness due to the length and complexity of that simulation. So if I have 1,000 individuals per generation, that's 1,000 times 10 minutes per generation just to evaluate the fitness of all of those individuals. And then on top of that, I might need to do crossover and things like that. So sometimes whatever little bit of computational time you can save will really help you ensure that your optimizer will end in a week and not a month. So sometimes it's just these savings. Or it might just be that the nature of the types of solutions you get out of here, they're a little bit smoother. And so you like that kind of averaging effect you get out here. Whereas with the pointwise crossover, you don't get averaging. So that's kind of, I guess, an interesting thing here is that with the pointwise crossover, if you want averaging between parents, you have to fuzzy it with mutation. You sort of require the mutation to give you something in between two parents. But with the linear combinations, the crossover naturally produces new results, which are in between parents, as opposed to just swapping things from each parent. So you kind of gain experience over time as to kind of, well, maybe I should try a different crossover here. So for now, we're just going to gain kind of exposure to the different options that you have when you're building these algorithms or using these algorithms. Questions about that? Yep.
Which one most closely resembles biology?
Well, so this is most inspired by biology. Two parents come in and they've each got, so like humans have got 23 pairs of chromosomes. And so when two humans come together, well, I could say this technically happens when sperm and egg are being made. And so you've got two pairs of chromosomes in each one of your cells. And so those two pairs come together and there is a point where they cross over to produce an egg, and in this case, two eggs and two sperm that will be a different combination of genes than the parent. So so this one, discrete crossover is what kind of happens here. So in biology, recombination does not change the alleles. It just changes the shuffling of the alleles. The only way to change the alleles is through mutation. And but we as engineers, we can introduce new ways to change the alleles. So this really becomes kind of an adaptive mutation where we are deciding to choose a totally new allele, an allele that is not in the population, based on the alleles we do see in the population. So mutation is sort of just like, we're going to choose something random. It might be around a parent, but it's kind of random. Here, we're saying we have two parents in this neighborhood, and we're going to create an offspring in between those two parents, or maybe away from those two parents, but on the kind of line that connects them together. So this is more artificial, but it's maybe more tractable, and it gives you a little bit more functionality. And it's just because engineers have built systems where we reliably can store real numbers. Biology, and part of that's because down in the depths of a computer, we do rely on discrete representations, but it's such high density, we assume that they're real. In biology, they haven't achieved that level of density. So when we view biology, we think of it discretely. So there's no analog to this in biology because really the genes are not, they're not as fine-grained as the discrete representations that we have. So we can approximate with this, whereas biology is kind of stuck with this. Okay, other questions on those two types of crossover? Before we actually, I'll show examples of what these crossovers do. All right, so let's do that. So in a linear combination case, then you've got two parents. So imagine here, I'm only worried about one gene. So this is like a parent with a single gene. So they're expressing a single allele. And so parent one is over here, call that mom. Parent two is over here, call that dad. So those two come together and they produce two offspring. Now, if this was mom and this here was dad, then the two offspring, Y1 and Y2, come from either doing alpha first or 1 minus alpha first. And so if alpha might be 0.2 and then 1 minus alpha is 0.8, then we're going to get 0.8 here and 0.2 here. So you just get these different mixtures. And so that's going to give you different weighted averages. And so if I look at the weighted average of X1 and X2, if I'm at the 75%, let's say this is 75%, then that will be, Y1 will be more like X1. So that means that I might be here because this is more like X1 than X2. But Y2 might be more like X2 than X1. So that's how, this is my like 0.25 X1 solution, and this is my this is my 0.75 X1 solution, and this is my 0.25 X1 solution. So I end up getting these two individuals which are in between the two parents. They're like interpolations of the two parents, weighted combinations of the two parents. Now, imagine this when you have multiple, in a full multivariate case. So now you've got these vectors that are in multidimensional space. And so, you've got this 3-dimensional, 4-dimensional, 1,000-dimensional mom and a 1,000-dimensional dad. And then now you're going to get an 1,000-dimensional weighted average between them, or two 1,000-dimensional weighted averages. And that's why we'll talk about convex or conical combinations, where you have to imagine a vector that's pointing in multiple space and another vector that's for you. So like if, I guess I might as well draw that. So if this were, a multi-dimensional space here, so I'll just do it in 2D. So if I have one parent here and another parent here, so this is parent one, and this is parent two, then if I combine the offspring together, I can get any vector in between these two. And so this might be, alpha equal to 0.75, and this might be alpha equal to 0.25. And so when we talk about the conical combination, what we're saying is that if we combine these so that we've got we're using these alphas between 0 and 1, where the weights add up to 1. So the weight on parent 1 is Gesundheit, and the weight on parent 2, those two weights add up to 1. Then when you add these two vectors together in 2 space, you can get any vector you want in between these, in this cone. Thus the conical combination, so it's a cone. And so imagine that, now do that in 3 space. So now you've got a third vector, and now you're looking at everything that's in between those 3 vectors. So that's what you're really getting by these mixtures, is that as you add more and more of these dimensions, you have to imagine sort of multi-dimensional cones where you're getting things inside the cone. The parents are constrained, or the offspring are constrained by the parents. So you can't go outside of the parents because alpha can't go any higher than 1. But you can swing around. You can be more like one parent, more like another parent, more like an equally weighted version of all of them. You can be the center of the cone, the edges of the cone, but that cone, you can't swing outside the cone. The ice cream's got to stay in the cone. But where you are in the cone, that's the so-called conical combination. And that's what we're getting here with this setup here. So we're getting all of these values here. Yep.
You measure the distance between x1 and x2? Would it be Euclidean or?
Well, so that's kind of you can measure the distance there, but it doesn't really matter to, like, this alpha is not a measure of distance. This alpha is just is how much you're mixing. You can measure the distance, and we can make statements about like transitivity or things like that. Like, the offspring is going to, it's like the triangle inequality. Like, we can say something about the offspring is sort of going to be, it'll be that x1, the parent 1 will be closer to the offspring than it is to parent 2. Parent 2 will be closer to the offspring than it is to parent 1. Those are sort of statements we could make about this, but alpha is not meant to be a distance, it's meant to be kind of a coordinate. Like, you could think about, maybe you could plot this on one of these like triangle things where you imagine there's parent 1, parent 2, and if you had a parent 3, but like we would in evolution strategies, then you could imagine that this space, like this would be a clone of parent 1, this would be a clone of parent 2, this would be a clone of parent 3, and then offspring that mix from those parents would show up at different points in this 3-dimensional space. And you could then talk about the points in this space. That's kind of looking down on this kind of cone. And you could talk about the coordinates of these points. Like, you could take this space and turn this into a plane where you just focus on where you are in this space. But I mean, that would just be kind of a visualization. So it's it doesn't affect the function here. But we were only worried about, for our GAs, 2 parents. And so we're just kind of worried about, as you swing alpha from 1 to 0, you're swinging it from a clone of parent 1 to a clone of parent 2 to mixtures of the 2 parents in between. Okay, so with that, for this 1-dimensional case, then you might have a fitness landscape. So this is our fitness function, which reflects our optimization objective. So our optimization objective might be minimize the loss of this neural network. And this would be the minimal loss of the neural network, the highest fitness position. So we've designed the fitness function so when you maximize the fitness function, you minimize the loss. But it turns out there's a local minimum here, which shows it was local maximum in fitness, and there's another local one over here. And mom and dad are close to these local optima. Now, what crossover does, this type of crossover does, is it ends up interpolating to create offspring that may be terrible combinations. So here it turns out that you have like a fraction of mom and a fraction of dad, and you get an unusable neural network. It's a really bad neural network. But it might just so happens that if you swap things in a different way, you might get an offspring that found itself on a new fitness peak. And that new fitness peak then, with more selection and mutation, can climb up and become an even better solution. So that's one of the things that crossover is doing here, is it's allowing us, so if we just depended on a little bit of mutation and selection, then these mom and dad solutions would never move in this direction, because any little bit of mutation would move downhill, and then selection would kill those. And so you only get uphill movements up to the drift barrier. So the drift barrier might set a cap, or anything inside here is practically equal fitness, but nobody's going to be moving down into this so-called adaptive valley. So one of the things that recombination can give you is a way to tunnel through these adaptive valleys, to jump from one fitness peak to another without having to ask selection to do the thing that is exactly the opposite that it's supposed to do. Selective pressure moves you up. So if you need to explore moving down, selection is going to work against you. But recombination, if you've got diverse parents, recombination creates an offspring that will really be a novel mixture, a totally new solution, which may find itself on a new fitness peak, which then selection can optimize. So that's the idea here. But I hope you see that if mom and dad are really close together, then your recombination, like if this is mom and this is dad, this is parent 1 and parent 2, then recombination will only allow you to explore this space. And so it really doesn't help you much more than like mutation would. And so it ends up being kind of counterproductive. So if you have lots of diversity to start with, recombination allows you to jump through the space in ways that mutation can't. But once you have little diversity in your system, recombination doesn't help. At least this linear recombination. But we're going to see this also applies to the discrete. Yep. Yeah, so the question is, how do we quantify diversity? There are metrics of diversity, like entropy, like just distance. Like you could just say, if you set some sort of like I know that in my initial population, there was a certain radius that all my things came from, that that I could draw, if I had my X1, X2, X3, these are my genes, and I drew my initial populations were inside some volume. Well, you could measure the volume, like, so your initials were samples out of this volume. Well, you could measure like the size of that volume or the diameter of that volume. And then it's costly, but then you can look at your current population and say, well, what volume do you live in? And you can see as things get more or less, that we know they're going to get less and less diverse over time. That's just selection and drift will make it so that you get less and less diversity over time. So you can measure diversity. And when we get to multi-objective and multimodal, we'll talk about ways that get a little bit more serious about trying to measure diversity and respond directly to it. If you do have a way to measure diversity quickly, you can use that to choose these alphas based on diversity, or choose mutation rates based on diversity, or choose the recombination fraction, the PC crossover fraction based on diversity. For now, we're just imagining they'll be constant. And because we know that diversity declines over time, a lot of people don't bother with trying to measure diversity. They just say, if I'm going to change, like if I'm going to turn off crossover, I'm going to kind of make another hyperparameter, which is like how many generations until I switch to the no crossover version. And you might just have to play with that. So you might say, well, I'm going to do 50 generations with crossover and then another 50 generations without. And I'm going to do a little pilot study and see if I start getting solutions that are consistently decent. And if they are, then I'll go with that. But if I notice that my solutions are very random and they don't seem to be very high fitness, one run they're this fitness, another run they're this fitness, then maybe I need to adjust my hyperparameters. So in theory, you can measure diversity and respond to it, but in practice, it's hard to know what's a good diversity, what's a bad diversity. All we know is diversity declines over time. Questions about that? Yeah.
Choosing the two parents as in similarity. So do they have to be like totally different? Because then they could be like it could be difficult to combine them, or do they have to be somewhat similar so that they kind of can combine the characteristics that naturally would go together? Excellent
question. So I'll talk about this briefly here in this lecture, but then we'll get to it. We'll return to it again when we get to all this stuff becomes more important for multi-objective and multimodal. But the question is like, is it can you always like, are there certain distances between the parents where you might have a better recombination success than others? And it turns out that there is, it's kind of like a sexual compatibility and they're actually in biology, there's also versions of this as well. You know, if you're within the same family, that's kind of like if you're very closely related here, then there are other things that happen in biology if you were to have mating within the same family. But even if there wasn't the stuff that breaks in biology when you have mating within the same family, you still have this issue that there's so little genetic diversity that you don't get any benefit from. But if there's too far away from each other, then you might say, like, I'm trying to build the fastest way to get from point A to point B. Well, you might have a cluster of solutions that have 2 wings, and you might have a cluster of solutions that have 4 wheels. And these 4-wheeled solutions have figured out a good way to drive fast on the ground. And these 2-winged solutions have figured out a good way to fly, maybe straight in the air. And if you do crossover between those 2 groups, you might get an offspring that has 2 wheels and 1 wing. And so that it's really low fitness. It's kind of like down here. And so there are strategies for choosing parents in the crossover that have some notion of similarity. Like you kind of take the dot product of the 2 parents and sort of say, are you kind of pointing in the same direction? And if in decision space, you've got parent 1 that is over here in decision space. So this is how many wheels it has, this is what fuel it uses or whatever it may be, this is how many wings it has no wings. And then you've got another parent which is pointing in this direction. Then they're not like pointing in the same 180-degree direction. So and so maybe their dot product comes up negative. So in that case, you set their crossover probability to 0. But if you had a third parent that happens to be over here, well, we'll allow those 2 to blend together. And so there are notions of species. So you can create these kind of quasi-species where within a population, you can say that we've gotten the origin of a particular species of solution and the origin of another species of solution. And we define species by genetic similarity, and we only allow crossover within species. So that's more advanced. It takes a lot more thought into how to do that efficiently, because now imagine you've got 100 individuals, and now you have to calculate similarity. That's an n-squared complexity. So every one of to decide if you just grab 2 at random, you could say I can calculate their similarity, but let's say they come up and they're dissimilar. Well, if I need a certain number of crossover, I still have to keep drawing them. So you end up getting this pairwise similarity. You have to build this network of similarity in order to do that, and that's pretty costly. So, how do you do that in a computationally efficient way? That's another kind of question. But in principle, yes, coming up with kind of a sexual compatibility approach is good because you don't want a lot of these solutions. Like if you just every crossover is a low fitness solution, then you may as well turn off crossover. So great question. It's just a more advanced topic. Yep.
So that goes in selection operator?
That would go in not the selection operator, but the how to create offspring operator. So the selection operator selects the breeding pool, but then we need to select breeders from the breeding pool to say who's going to produce offspring by crossing. And so I would call that, it could go in the crossover operator, but it's almost a special thing. It's before you call crossover. It's sort of a parent selection. So it's like another layer, an in-between operator that we haven't, so in general, we have selection, we have crossover. But what we're talking about here was like for a much more sophisticated GA, having something in between those, which only selects kind of likely productive parental pairs for the crossover. So it's kind of before we had a crossover fraction, we're sort of making the crossover fraction dependent upon the pairing. So it's like taking that one hyperparameter that was a static and turning it into a function. So it's like, all right, now I'm going to go through every possible one of the n-squared pairs and then calculate the crossover probability for that pair. And for this pair, it might be 0. For this pair, it might be 99%, depending on their combined properties. Great question. All right, well, let's Great, keep those in mind, and like I'm just sort of for progress, I'm going to keep rolling here. So we go into point-wise crossover and say, okay, we can maybe understand the linear combination case, but what's going on with the point-wise? And it's basically the same sort of thing that's going on in linear combination. So the idea here is if I have two parents, I went over this example before. So here's two parents, and their genes are the place values. So this one has allele 1, allele 2, allele 3, allele 6, allele 0. And this one's allele 6, allele 1, allele 2, allele 0, allele 5. And so the place values here in this decimal representation, those are the genes. So for these two parents, I've selected randomly this point to do crossover. So I'm going to keep these the same and swap these. So when I do that, these are the two offspring. These were kept the same, these were swapped. So if you look at it, when I swapped them here, these offspring are now genotypes which are in between these two genotypes. This is basically looks just like that linear combination where that alpha was less than 1. It created intermediate solutions. But if I happen to have choose this crossover point, if I swap these, then now I get offspring which are farther away from the two parents. So these parents went from 0.6 to 5, and now the offspring go from basically 0 to 5.6. So the different crossover points, which are selected at random, sometimes interpolate between parents and sometimes extrapolate from around parents. They're still borrowing from the parents, but they're basically doing you could come up with if for at least a numerical case, you could come up with a mathematical operator that would tend to do the same sorts of things. So this is how we conceptualize what discrete point-based crossover is doing. Is it's basically doing something we view as a linear combination. That's part of the reason why a lot of people just say, I'm not going to bother with making this ugly crossover operator that has to choose and swap and all that. If all these are numbers, I'm just going to do the linear combination because I can come up with ways to do linear combinations that give me the same outcome. Now, by looking at this here, any questions about this? Yeah.
So when we say this interpolation and when we say extrapolation, because I can see on the above, there are certain points like 5.032 is where it's above 5.02, but still we goes on interpolation.
Right, yeah, well, I mean, this is my interpretation. So I was just trying to show, I was mainly focusing on this one, and I probably could have come up with a better example. Well, I was just trying to say that I was hoping that what this would motivate, and maybe I can change the notes here, is that when you swap digits of a representation, what you're basically doing is creating offspring that are interpolations and extrapolations. So here you can say, I've got both. I got an offspring that's in between and an offspring that's a little bit farther out. And here I've got two offspring that are both farther out. That's what I was trying to communicate. I just squinted, I guess, when I wrote this, and I could have come up with a better example. So apologies for that, but hopefully that's kind of clear there, is that you can get these, you're still mixing from the parents. The offspring are kind of related to the parents, but they're related in sort of an in-between or on the same line as the parents. And so that generates these new offspring. Now, if the parents are really similar, so here we've got, and I've got a question like this on your mini project. You've got parents which are similar up to the fourth digit of their representation. So they only differ down here in these the least significant digits of the representation. So at this point, if I were to do crossover here, I would get exactly the same offspring as the parents. I mean, it would swap, but I'd basically just be swapping the identities of the two. So I wouldn't actually be creating a new offspring because these bits or these digits would go here, these digits would go here, and now the two offspring would have the same, no genetic difference from the two parents. They just happen to switch their position in our indexing, which doesn't matter. I only get differences if the crossover operator happens to draw a line here. Then at that point, I break up the linkage between 4 and 2 and a 9 and 6. So I get a 4, 6, and a 9, 2. That's the only time I end up getting any differences between these. So this goes to show that as your genetic similarity gets less and less and less, the effect of crossover gets less and less, and you can end up doing the same thing or better with a small degree of mutation. So if I had a small mutation operator, I know we haven't formally talked about mutation exactly yet, but it would be easier for me to add a little bit of noise to these parents to produce noisy offspring than to swap them. Because at this level, I'm fine tuning. I'm not looking for qualitatively new solutions. These are not qualitatively new solutions. These are attempts at fine tuning, and recombination is not a good fine tuning operator. So this is an example of how crossover kind of naturally turns itself off over time. But if you're like in MATLAB you have a crossover fraction where those individuals only do crossover, and another fraction where those individuals only do mutation. Well, at this point, those individuals that only do crossover, they're just going to clone the parents. So now you've just got an additional numerosity of the parents, and that might not be what you want. So as the diversity declines, you might want to shift towards mutation, maybe with a small amount of noise, because crossover is just kind of wasting time. It's just duplicating the parents. So so with that, crossover is not usually as effective as mutation. You often turn it off, set the probability of crossover, the crossover fraction to 0. That can open up more mutation slots in algorithms like MATLAB's. But then, as we already just mentioned before, if you have really high diversity, you might want to be careful about which parents you cross over, because if they're very different, it's like if you've got a neural network, it's got millions of weights, and you've got one neural network that happens to, like, there could be a cat-dog classifier. There could be one neural network that has a huge number of weights. If all it's doing is classifying cats and dogs, there are probably a bunch of different ways to configure the millions of weights of that neural network to classify cats and dogs pretty well. And so you might be under one of those local optima for that classifier, and then you might have another population, another local optima for that classifier. If you recombine those two neural network weights, you won't necessarily get a classifier that's better. It might be terrible. And so you might want to just be doing recombination among things that are sort of focusing on the same internal latent. We haven't talked about neural networks, but feed-forward neural networks, they develop latent internal representations of the world. And if the internal representation of the world in this cluster is very different than the internal representation of the world in this cluster, then doing a crossover between them might just produce a really confused neural network. Whereas crossover here, then these have the same internal representation, so that's more likely to produce something that still works. So that's kind of what we're saying here. So you turn it off when the genotypic diversity is really low, but you might also prevent it from happening when the diversity or the difference is really high. All right, questions about this? So there's a sweet spot where your crossover works really well. And the easiest thing to do is to just turn off crossover after a certain amount of time. And so if you make a lot of bad combinations, you're going to burn fitness evaluations, which may be costly. It could be 10 minutes of fitness evaluation, but at least they'll have low fitness and they'll be purged from the population. And then eventually you'll turn off crossover after like 100 rounds or 100 generations or 50 generations, and then you don't have to worry about this because crossover's done. All right, so then the last operator, mutation. So this is a little easier because we don't have to deal with one parent at a time. And so we're going to basically create offspring that are asexual, so they're based on one parent. And they're going to be perturbed versions relative to the parent. So each of n genes will be mutated with some probability. That's your hyperparameter. It's the probability that a given gene will be mutated. So in MATLAB, that the number of offspring that are populated by mutation is the so-called mutation fraction, and it's just 1 minus the crossover fraction. So if you said I want 75% of the offspring by crossover, then the other 25% would be by mutation. In the book, then every one that gets crossover also gets mutation, or basically every offspring gets mutated. You can turn off crossover by affecting the kind of crossover rate, but you're still going to produce offspring, and every offspring then gets mutation. Okay, so what do mutation operators look like? The simplest mutation operator, basically going to go over 2 mutation operators. The simplest mutation operator is the uniform mutation operator, where you just go down through every gene. So you've got a parent, and you said, this parent is going to go through asexual reproduction. So I'm going to then populate one of my offspring slots with a mutated offspring. So I'm going to copy that parent into the slot and then mutate that slot. And what I do is I go down through every gene of that parent. So this is one gene of that parent, parent J, gene I, and I'm going to then draw uniformly among all the possible alleles for that gene. So I'm not paying attention to what allele the parent had. I'm just saying at that gene, the parent might have had, this might have been the number of wheels on the car, and the parent might have had a 3, and I'm just going to draw and say, well, I could have anywhere from 1, 2, 3, or 4, and so I'm going to pick randomly with 25% probability of each one of those. I might end up getting the exact same thing the parent got, or I get something different. So that is uniform here, and this could be continuous or discrete. You just draw from a full range on the individual J of gene I. Okay, now, and the point of all of this is to just try to maintain genotypic diversity, because again, remember, selection and drift are purging diversity, and gradually all of the solutions are looking more and more alike, and mutation is just trying to add more diversity. Okay, the other way I could do it is non-uniform, what I'll call additive mutation, where here I actually do focus on whatever the allele the parent had, and then I perturb it by a little bit. And so this could be a uniform, but it also could be normally distributed. But the idea here is I'm adding a little bit of noise. Maybe maybe negative, maybe positive, but it's zero mean white noise. So so white meaning that it is independent of whatever the previous draw was. Zero mean, so that I've got no bias in which direction I'm going. And then I just basically perturb it by that amount. If this had 3 wheels, then maybe I perturb it by 1, so it could go down to 2 or up to 4. And so I can do that. Like I said, I can also use a Gaussian, and I also can change W over time. So early, W could be a hyperparameter, which is the initial W, but then there's a schedule of how it decays toward the end. So you might say, I'm going to run 100 generations. Well, every generation, maybe I decrease W by 1%. So then over 100 generations, I turn off mutation in the very last generation. So that's a way to shift from mutation for exploration to mutation to exploitation to just turning mutation off. And so that is very common to come up with a schedule of turning that mutation rate down, similar to having a schedule of turning the crossover rate down. All right, so questions about that? Mutation, you're just fuzzifying the offspring. You're just adding random noise to the offspring, either really random noise, uncorrelated with the parent, or you're just perturbing the parent by certain values. Yeah.
The difference between mutation by exploration, exploitation, and then zero, like those, like what is the thing that we say it's exploration and then it's exploitation?
That's a good question. So it kind of goes down to it's a little subjective, but if we go back to our fitness landscape, and let's say my fitness peaks are spaced this way. So I would say that exploration would mean you have a W, which could get you to another fitness peak. So we'll call that exploration. But then exploitation might be a W where it will keep you on, I guess I should make this clear that it's exploit versus explore. So if it's a big W, so the thing is, you have to know a little bit about your optimization objective. And so you have to have some sense. This is very Bayesian, right? You have to have some prior idea of how big moves you want to make. And so this noise factor, if they're, you kind of want to tune this to how far away you think qualitatively different solutions are. And that will give you exploration. But then as it shrinks to be smaller than a fitness peak, then that kind of guarantees that you're going to do fine tuning on that fitness peak. And then why might you bring it all the way to zero? Well, you just want to try to, if you bring it to zero kind of slowly enough, then that helps ensure that you're going to keep kind of climbing, and then maybe you'll get lucky enough that when it stops, right at the last generation, you'll be right at the top of that peak. That's the hope there. So questions about that? Yep.
So in non-uniform mutation, the additive value need not be discrete, right?
No. This uniform. It's a great clarification here. This could be discrete or continuous, and then likewise, this is discrete or continuous. So it has to be consistent with the gene. So consistent with what the allowable alleles are. Normally, when we think about doing non-uniform mutation, it's usually on a gene that is a real number, so that we're pretty safe just by drawing a random number and adding to it. But you could do additive mutation on discrete genes as well. You just have to be careful that as you're perturbing them, that you come up with something that is still feasible.
So the way we are encoding the gene is we are having like individual discrete numbers and then we are representing it as a decimal value.
Well, that's up to you when you do the GA. So that was my example back here for crossover, just to illustrate that. And for mini project 1, I asked you to do one in this way. And I asked you to do it 2 ways. So I think in question 1 on mini project 1, I have you encode the gene this way. And then I have you compare it to one where this 1, 2, 3, 4, 5, 6 genes turns into one gene that is real valued. So that is completely up to you how to do that encoding, and that takes some sort of trying to think about what you want the GA to do and what the problem is like. But once you make that choice, then when you choose the mutation operators have to be consistent with that choice. So as an example, in the mini project, there will be constraints on these where it can't be higher than 1.5 or lower than negative 0.2 or something like that. Well, then then the constraints on these digits are going to be coupled. So if you're positive, then this digit can go all the way up to 1. But if you're negative, this digit can't go any lower than 0 or any higher than 0. So, that you have to be careful when you're mutating, that you're mutating within the valid range. Other questions? Hopefully that's a more straightforward operator than the other operators. You're just adding noise. Okay. All right, so bringing that all together, all 3 of those operators, all 3 of those types of operators, and they can be applied just as much to the GA from the book as the GA from MATLAB. So even though, again, the population structure is different, then you still can use the same operators. You're still making the same types of choices. So, but how do you choose all of these? We've got a lot of hyperparameters. We've got M, R, and E. We've got PC. We've got PM. We've got all this selection pressure stuff. So, and then I wanted to add briefly here, we've talked about roulette wheel selection. So that's the classical way to teach this. But now when you do roulette wheel selection, there's an alternative that you see instead called stochastic uniform sampling. And so this reduces variance in the roulette wheel selection. So modern GAs, Even though we talk about them in the old textbooks using roulette wheel selection, modern GAs often use stochastic uniform sampling. Now, how does that work? So you've got your probabilities that are proportional to fitness, like you would in fitness proportionate selection. So you can place those on your interval, like in fitness proportionate selection. So this is individual one's probability because they're higher fitness than individual two. They take up more space than individual two. Individual M has maybe really high fitness, so it takes up the most space, and so on. So you've got those all stacked up. So all these things add up to 1. So in roulette wheel selection, you draw R random numbers between 0 and 1, and you ask, and you see where they hit. Maybe the first number hits here, and then that means you're going to draw parent 1. Maybe the second number hits here, so you'll draw parent M. Maybe the third number is back to parent 1, the fourth number parent 2, and so on. But you draw R numbers, and you see where they land. Stochastic uniform sampling it does it a little differently. In stochastic uniform sampling, you draw one number, a single number, and you draw not between 0 and 1, you draw it between 0 and 1 divided by R, where R is your number of parents. And that will land, somewhere early in this. So then you're going to then add 1 minus R minus 1 times. So did I say 1 over R minus 1 times to give you the other points. So the first point will land low in the interval. The second point will be 1 minus R away from it, 1 minus R away from it, and so on. So each one of these will be not 1 minus R, 1 over R away from each other. So the first point is a random draw from 0 to 1 over R. And all of the other R minus 1 points are just 1 minus R displacements from that. And that will give you R points in this interval. But it helps sort of guarantee that you're not going to accidentally, it reduces variance and that you don't get a pileup of samples in one region. It sort of helps spread these things out in an honest way. So this is something that this is what we call in simulation. So if I've taken any simulation courses, this is an example of a VRT, a variance reduction technique. So it's just a different way to get the same output where the average behavior is the same, but the variance is different. And so if you've taken any statistics courses, maybe design of experiments courses, maybe a little bit of machine learning courses, you might have learned about stratified sampling. So there are ways in which we process large amounts of data and we try to if you're familiar with an ANOVA, there's a non-parametric version of ANOVA called a permanova. And a permanova does stratified sampling in order to reduce variance when you're trying to figure out the difference between a bunch of different groups. Well, this is a version of the so-called stratified sampling. So you're drawing one number and you're pushing all the other numbers. So you're basically saying, I'm creating strata, which are 1 minus R wide. And then I'm sampling within those strata. So, how does this relate to like real world sampling? Well, this would be like, rather than sampling the whole population, let's say I know that there's 55% women and 45% men. Well, I might sample men and women separately, and I just make sure that 55% of my samples come from the women group and 45% samples come from the men group, and then I pull those together afterwards. That's an example of stratified sampling. If I just sample from the whole population, I can get a lot of variance because I might happen to get a disproportionately high number of men or a disproportionately high number of women. So by putting them into strata and then sampling within the strata, then I reduce that additional variance. And that works well here. And by reducing the variance, then this lowers the drift barrier. It provides more ability for your selective pressure to act. So that's a reason why it basically doesn't change the 1/r structure of the drift barrier, but it changes the scalar in front of the 1/r that sets that drift barrier. So it allows for a smaller selective pressure to work because you're reducing the variance in the selection. Your question first.
Aren't you increasing the variance in the selection of the children?
No, because you're creating these 1 minus r strata, these layers that are each 1 minus r wide, and then you're drawing within those layers. So if this is the male or female solution, there might, you could imagine these are the males in the population, these are the females in the population, rather than, I want to sample like height or something, or I want to sample, I don't know, A1C level, in the blood or something like that. And I could just pick 100 random people, or I could pick 55 women and 45 men. And then, and that helps ensure that I don't get a disproportionate bias from just random luck. That, like, if I were to just draw 100 people randomly, I might get 70 men and 30 women. So that's the idea here, is that you create these strata that cover the whole 0 to 1 interval, and then you sample within those strata.
That would be increasing it, right?
That decreases the variance, because it's if you're talking about which parent you choose, then it makes it less likely for one parent to. So right now, if your parent won, there is a chance that you could be picked R times. And if your parent, M, or any parent has a chance they could be picked 0 to R times. But by creating these strata, you reduce the variance in how often parents can be picked. Because it might be that this parent stretches across, a large area, so it happens to hit 4 of these stratified layers. Well, that parent can now be picked 4 times or less. Like, it reduces the number of times that a single parent can be picked. And it keeps them closer to the fitness proportionality. It helps to guarantee that no parent is, just by good luck, going to get way more representation than their fitness suggests they should get. That's why
It's badly worded, that's all.
it's worded the way it's worded. Because that's the right way to word it.
So the VRT usually reduces the variance over the experiments, like when we use the stratas. So what I'm thinking here, is this reducing the variance over the generations? Because VRT usually don't work in a single experiment.
It is reducing, so you have to think about it as like, the variance you're reducing is the number of individuals that will be drawn for a parent. So what we're saying is, regardless of whether you use roulette wheel selection or SUS, SUS, the average number of parents will be dependent upon fitness. So if you have 50% of the fitness, you will have 50% of the offspring. So if there's 10 offspring, you'll get on average 5 of them in both. But for that one draw, so that would be even with, so when I guess I'd say for that one generation, when you draw all of the 10 spots, if you were to do that over and over and over again, then for one selection of those 10 parents, then you're going to reduce the variance in in the individual level, the variance around that average. So on average, I should get 5 offspring, you should get 1 offspring, you should get 0 offspring. That's going to be the same for both. But if I do my selection operators, we do all of our offspring. We do that 100 times. We can do the variance in that 100 population, and I'll see that under SUS, I will be very much closer to my 5 than under roulette wheel, where I could go all the way to 0 or all the way up to 10.
So basically the sampling mean of, let's say 100 generations will converge to a similar value over the iterations.
Yeah, right. For if you do lots and lots of generations, the mean for both of these will be identical. All right, so I don't want to keep you guys much longer than that. So next time we'll finish these 2 slides, which I'm going to talk about exponential ranking is another modification. And then I want to bring together this graph, which pulls together all 5 of those parameters. So we know of how does one parameter trade against the other parameter when you're tuning your genetic algorithm. So, and then after that, we'll go into unit 2. So, all right, that's it. So it's a good discussion, a lot of great questions. And this will only take a couple of minutes at the beginning of next time to finish this and we can start unit 2. But because of that, I mean, just because it's so fresh in your mind, we got through mutation, so technically I could still make the everything due Wednesday. I'm just going to push it to Sunday. So I'll push everything a couple of days again. So everything that was due tomorrow will be due Sunday. Everything that's due next Wednesday will be due next. Thank you. All right. See you on Thursday.